Okay, welcome everybody. This is workshop number 20, single cell RNA sequencing analysis using R. My name is Giovanni Quiñones Valdez. You can just call me Gio. I recently graduated from UCLA with a PhD in bioengineering, and my research focused mostly on the analysis of RNA sequencing data, different types of RNA sequencing. And we're using this type of data a lot to answer different questions about genetic variants and other nucleotide variants present in RNA, and how these variants regulate different aspects of RNA processing.
You have all these different niches, right? Within the team.
I believe somebody's mic is on. There is a little bit of background noise, so please keep your mic muted. So I wanted to go a little bit over the structure of this workshop. First of all, I think it's worth highlighting that this is an introductory level course on single cell RNA sequencing data, which means that I'll try to cover as much of the pipeline as possible, as much of every single step and everything that goes into obtaining this data, analyzing it, and then making some meaningful biological observations. So at this point, I wanted to make a little poll. Based on my roster, I see most of you have affiliation with either School of Medicine or Life Science. So can we maybe do a show of hands? Who of you have some background in biology or life science? Okay, I think that should be most of you. Okay, that's great. And who of you, let's do another show of hands, who of you have some experience with programming? See, show of hands if you had any experience with programming. Okay, that's great. Yeah, so one of the prerequisites for this workshop is the intro to R. And like I mentioned in my email, where you are not going to be writing any code, any line of code. The code is already written for you. You'll just have to run it on your own computer. So you don't really need any advanced knowledge in R, but it's still good that you understand the syntax and you understand what the code is doing, just in order for these things to make any sort of sense to you. Another point that I wanted to mention is some of you are taking this workshop for credits for the BioInfo 275 A and B. Can we have a show of hands? Who of you are taking this for credit? I believe there should be a few. Is nobody? Okay, well, that's really bad because participation and attendance is a very big part of your grade. Hopefully they will show up soon. And also, if you're taking this course for credit, you'll also be assigned homework and a quiz, which they will be graded and will assign you a letter grade based on these assignments. If you're not taking this workshop for credits, you can also do the homework in order to test what you learn. I'll be happy to give you feedback. And the last point is that attendance and participation is required. So we really intend for these workshops to be interactive, for people to participate as much as possible. So please turn on your cameras if you haven't done that. I also kind of want to, it's also nice to see you're actually talking to an audience and not just a screen. So yeah, please turn your cameras on. I want to see as many students as possible. The structure of this workshop in terms of material, we'll be going over, this workshop is 3 days long. On the first day, what we're going to cover are some concepts in single-cell RNA sequencing, some concepts in the experimental design, and we're going to conclude with some data exploration. So today is going to be a little more on the theory side of things. We're going to learn some facts, some of the pros and cons of single-cell RNA sequencing. And we're also going to take a look at the different types of data that we're going to be working on. On the second day, we're going to talk about quality control, data processing, clustering, and visualization of single-cell RNA sequencing data. The second day is going to be more hands-on, where each one of us are going to run the code in order to process the RNA sequencing data. We're going to make some plots. We're going to look how the different cells cluster together, and we're going to explore what can we learn based on this analysis. And lastly, on the third day, we're going to continue with the topic of clustering, because it's very extensive and there is really a lot of things to mention on this. So whatever we do not manage to cover on the second day, we're going to finish on the third day on clustering. And we're also going to talk about some very interesting analysis that can be done with this type of data, which is the pseudo time. And this is one more on the more modern techniques that people have developed on the analysis of single cells. And it's really become a focal point in this field, because this pseudo-time trajectory is actually the method by which researchers learn a lot about lineage of tissues, differentiation of cells, and obtaining cell lineages and so on. So we're also going to be talking about pseudo-time estimates. And lastly, I wanted to bring up a little more broader topics, not so much on the technical side, but rather more on the critical side of single-cell RNA sequencing, meaning here in what I call perspectives and future directions, I wanted to mention certain, for example, limitations of this type of technology. How are people using them? And what are researchers looking forward in when it comes to newer technologies, newer implementations? Pretty much have a more critical view of this technology in order to make a complete assessment. And the learning objectives for this workshop, first of all, is to understand the motivation behind the sequencing of single cells. So I believe that's a very important question to understand. First of all, why do we care of single cells in the first place? So being able to understand the motivation is going to be one of the main objectives here. The second one, to learn about the steps and tools that we can use for data collection, processing, and analysis. Of course, at this point, there are hundreds, probably thousands of different tools. People develop new protocols, new technologies, new pieces of software every day, and it's virtually impossible to keep up with everything. But however, I wanted to introduce you to the most common ones, the most useful ones, in order for you to make comparisons and to know what each tool is used for. And the third objective is, of course, not just to know the tools, but to also use them. So being able to analyze the data and interpret the results. And lastly, the last point I mentioned before is to learn about the perspectives and future directions of this technology. That's the part that I mentioned, having a more critical view, knowing in which direction the field is moving, what are the limitations and what are the things we're still missing. So we're going to get started. This is the plan for today. We're going to start with the concepts in single-cell RNA sequencing, experimental design, and towards the end, we're going to do some data exploration. Right. I'm sorry, I was just reading some question in the chat real quick. If you have, when you're done, you shows the integrations. Right, so in the third date, as part of the, somebody's asking about data integration. So we're going to talk about that in the third date. I prepared some material in order to talk about how to handle different batches, different replications of when you design an experiment. So going back to the slides, today we're going to start with the central dogma of biology, which if you are in the life science or you studied biology or biology related a biology related major, you've probably seen this diagram 1,000 times. So I'm going to show it to you for the 1,001th time, just to give you a quick overview of exactly what we're talking about here. So as you know, the genetic information flows from DNA into RNA and into protein. The DNA, the analogy that we're using here is the DNA is the hard copy of your genetic information, RNA, the soft copy, ribosomes being the 3D printers, and the protein, the functional part, is the 3D printed object. So the reason why we're starting from this point is that I want to show you that we're targeting this particular molecule, the mRNA, which is the intermediate step in the central dogma. And the process of RNA sequencing pretty much means the sequencing, the reading of this RNA molecule. We're trying to see exactly what this RNA molecule looks like. So what does it mean to read the molecule? What information are we gaining? We're learning quite a few things actually when we sequence RNA. We're learning, for example, where these molecules come from, which means the gene of origin. So we know that for every particular gene that is encoded in your DNA, through the process of transcription, you're going to generate multiple copies of RNA. And thanks to sequencing, we can collect these molecules and being able to find out which gene they came from. We can also look at how many of these molecules are coming from a particular gene. And that is what we know as expression. So if a particular gene is generating 100 mRNA copies, let's call it gene A, whereas gene B is generating only 2 copies of mRNA. Therefore, we can conclude gene A is having a much higher expression than gene B. And thanks to RNA sequencing, we can learn this type of information. And we can also see exactly what the RNA molecule looks like in comparison to the original DNA sequence or in comparison to our reference sequence. And this is what we'll call the different variants that may be present, meaning the differences in sequence, or also a lot of RNA molecules are being processed. So through process such as splicing or RNA turnover, decay, polyadenylation, and so on. These different processes are going to change this soft copy. And thanks to RNA sequencing, we can see exactly what these changes are. Right. So traditionally, this process of RNA sequencing has been done in bulk. Now, what does it mean to sequence RNA in bulk? In this diagram, we can see that we'll usually start with some tissue. And as you can see, a typical tissue will be made up of different types of cells. And the way bulk RNA sequencing is typically performed is that you'll obtain all these cells, you will purify RNA from them at the same time. And eventually what you'll get is with a pool of RNAs that come from different cell types, different individual cells from the same tissue. And once you obtain this mixture of RNAs and you process them, sequence, and get some data, you'll obtain a signal. And this signal, in this case, gene expression, is the average signal for all the cells that were present in the batch. And although this technology is being used for more than a decade at this point, and a lot of studies have been published using bulk RNA sequencing data, it presents one very significant limitation. And that limitation is that from the signal that you obtain, we cannot learn anything of the original cell population. The information about the original cell population gets completely lost in the bulk analysis. So enter single cell RNA sequencing, which is the process by which we first try to isolate individual cells that come from this tissue, and then we analyze individual cells separately. In this diagram, we can see that for the different cell types, we obtain some features, some data that is unique and different across cell types. And based on these differences, on these different profiles, we can reconstruct what the original cell population was in our tissue of origin. So one way we can quickly summarize this is that bulk RNA sequencing obscures cell-to-cell variability, and single-cell RNA sequencing, the whole point is to obtain individual cell information and explore this variability across cells. If at any point you have any questions, please feel free to raise your hand. I'll try to accommodate as many questions as I go by. I'll keep an eye on the chat box also and on the screen, but if I do miss your hand, also please feel free to interrupt. Now, the study of individual cells is actually was actually much needed, and it's actually something that's been pursued for a very long time. If you may wonder, what is really the advantage of getting a signal for every individual cells as opposed to getting an average signal for a tissue? So in order to illustrate this, I put up this image from this paper, which it actually is presenting what is the composition, the histology of a tissue. And you can see that you start labeling different parts, different sections of this tissue. And then when you zoom into a very small section, you see that the composition of cells is actually very heterogeneous. You'll see that within a very small cluster of your tissue, you can see that you have very specialized cells. Cells that their morphology are very different from each other, but not only their shape, also the function, the amount of genes and the genes and the amount of genes that are getting expressed within each cell is different to each other. And you can see that it's not, when you have a collection of very specialized cells, it's no longer biologically meaningful to get signals, one data point, one signal that represents this vast diversity of cells. So that's the point, that would be the main motivation for single cell analysis, is that within a tissue, we can observe multiple types of cells, and each one being highly specialized. Each cell has a unique transcriptomic fingerprint. That means that, like I mentioned, the genes that are getting expressed and by how much they're getting expressed, that will also be unique to individual cells. And probably the most important point also is that in a typical study that compares the reaction to a specific treatment or condition, this treatment or condition may affect only a specific type of cell. So say whatever condition, whatever stimuli you're testing for affects the green cells. So in order to identify these changes, it's important to get a signal that is only for the green cells. And when you perform the study in batch, actually the signal is going to get lost because you are mixing it with the signal from the other cells. And this is also something that's been reported in the literature before, that many cells show very specific features such as allele-specific expression, meaning when only one of your copies of the gene gets expressed and not the other. But because this was only happening on specific cells, and the study was done using bulk RNA sequencing, it was impossible to detect this pattern because it was getting hindered by the signal from all these other cells that did not have allele-specific expression. Yes, there's a question. Kwanji. You may need to unmute your mic. Okay. You can feel free to write your questions in the chat also. Maybe you just forgot to lower your hand. So some of the applications of this technology is, there's another question. Yeah, Christopher.
People have their hands up from when you did the poll at the beginning. I think it's just people. Yeah.
Oh, sorry. I'm sorry. What was your question?
That was unintentional. Sorry about that.
Okay. Yeah, let's carry on. So some of the applications of this type of technology is to study and identify cellular heterogeneity. This means to identify the different cell types, cell populations within tissue. To discover new cell types. And this is a very interesting point though, because this is actually what motivated the study of single cells in the first place. I believe in the 90s, people wanted to catalog all the different types of cells that were present in the brain. So they pioneered methods to get gene expression signal from individual cells. To discover new markers and regulatory pathways. So through the analysis of single cell RNA sequencing, as you'll see in much more detail tomorrow, you'll see that what makes cells different from each other is this transcriptomic fingerprint, meaning the genes are getting expressed. And so while trying to identify new types of cells or new cell populations, you also run into a particular set of genes that make this cell unique. And this particular set of genes may be involved in a regulatory pathway, or by definition is what we'll consider a marker genes. Genes are used to identify different cells. To reconstruct cellular lineages, this is what I mentioned briefly about the pseudotime, which is trying to identify the phylogenetic connection of different cells, meaning how one type of cell may derive from another one. And this is something that we can also explore using this type of data. So let's go through a little bit of history. In 1992, Everwine et al. Measured the expression of a handful of genes from single cells using in vivo reverse transcription and in vitro transcription. So what happened in 1992, and that's what I was mentioning before, is that this group tried to identify the different cell types that are present in the brain. And the way they were doing it back then, in order to identify individual cells, was to manually dissolve the cell suspension and do multiple rounds of dilution until you get to a point that statistically you either have one cell in your tube or 0 cells. So as you can imagine, that takes a lot of manual work in order to eventually come up with a concentration where only one cell may be present. And then they perform some targeted amplification using in vitro transcription. We'll mention what this is exactly later, but it's just a set of experiments that will try to amplify the signal of specific genes. So as you can imagine, these early studies were very limited to just a handful of genes. I believe it was 10, 20 genes, something like that, and for a few cells that they were able to collect. Starting on 1992 to 2004, this method was greatly improved thanks to the advances made on PCR. Hopefully, most of you are familiar with what PCR is. In just a few words, is a technique used in the lab to amplify the signal from either DNA or RNA via conversion to cDNA. And these PCR-based methods actually were able to increase the throughput of these experiments. By the use of primers, you were able to target a lot more genes, and thus increasing the capacity of these earlier experiments. And this is something that was predominantly done up to 2004, roughly, because in 2003, we enter a new era where a different technique is being used. And that technique is called microarrays. To very briefly explain what microarrays are, this is a high throughput technique where you insert DNA aptamers that will target pretty much all the nucleic acid, well, in this case, all the RNA from your samples, and amplify the signal via fluorescence. And another improvement or difference of this technique is that it was untargeted, meaning that people were no longer looking at a specific set of genes, but they wanted to amplify as many of the genes that were present as possible. And what happened after 2008 is that in 2009, this group of Tang et al., they adapted a method that was present. This method was high throughput DNA sequencing. High throughput DNA sequencing is using very advanced sequencing technology, but at that time was used only for amplifying genomes, amplifying DNA. So what this group did was to adapt this protocol in order to amplify the signal of mRNA from cells that they were able to isolate manually. So in 2009, we have our earliest example of single cell RNA sequencing in what we know today using the sequencer technology of bridge amplification. And the way these improvements throughout these years looks, I was going to say history, but it's actually, it actually just happened within the last 20, 30 years. But anyways, within this last, between 1992, where the signal from the first cells, first individual cells were obtained, up to 2009, where we get our first experiment that used a sequencing technology by Tang et al., we see that that group was able to amplify signal, to obtain signal for, the very first paper was only one cell. But then in the next year, they were able to produce a few dozens of cells. And you can see that throughout the years, in the last 13 years, the amount of single cells that people were able to sequence has been increasing exponentially. You can see our Y coordinates are in the log scale, to the point that in 2017, one of the latest technologies, for example, 10x genomics, was able to produce over 100,000 cells. And this graph is from a paper from 2018. Actually, later on, using 10x genomics, people were able to pass the 1 million mark by being able to sequence over a million. I believe it was 1,300,000 cells using 10x genomics. So you can see this very steady increase on the throughput, on the number of cells that we're able to obtain signal from. And some of the things that we can mention here is that this STRT sequencing is one of the first commercial protocols that were developed, and so goes for Cell-seq and Smart-seq. And these earlier methods were still using a bunch of manual approaches in order to isolate and get individual cells. And then later on, where we have Fluidigm and then Mars-seq, these methods were already using more advanced technologies to isolate the single cells, which I'll also mention later. But I think it's important to get a picture that starting from this point, roughly 2013, 2014, people started using microfluidic technology in order to automate the isolation of cells, and thus being able to sequence much larger numbers. So we're going to go over the experimental design. This is pretty much the outline of how cells go from our tissue of interest all the way down to this type of plots, this type of analysis that give us information about the different cellular, even about the different cell populations. So how do we go from our tissue of interest all the way to obtaining some meaningful biological signal? The different steps are outlined in this slide. See that once we have our starting tissue material, the next step is the dissociation of cells. This step consists on, because in the tissue, cells are very interconnected. They contain all these molecular bindings between cells that you have to break apart in order to get a suspension of cells. So you can have individual cells floating in solution. So this type of dissociation is done through a number of different ways. So a lot of times people use enzymes to break some of the extracellular components of the cell in order to dissociate them. People also use different types of charge molecules like cations in order to create some magnetic, some binding of the charge ions in order to induce the hydrolysis, the separation of these molecular bindings. Another approach is using chelating agents, also that when you incorporate it in solution, it will result on the separation of these extracellular proteins. And once you obtain your solution or your dissociation of cells, the next step is very critical, and this is where the different technologies come in place, which is how do you isolate individual cells? How do you come up with a series of steps that you guarantee you're going to sequence cells individually? And here we're going to go over some technologies, some examples of how this is done. I think for this slide, it's just important to pinpoint at this particular arrow as this being one of the most important steps in the experimental design. And once you isolate your cells and you have an individual cell from which you want to get signal, you extract RNA. Now, RNA extraction is done, very briefly speaking, through a lysis of the cell wall, and then using a combination of phenol chloroform in order to extract the RNA, to purify the RNA molecule, which is negatively charged. This step, is mostly chemistry. We're not going to get into the details of the chemistry that takes place. So we're just going to briefly mention how the RNA is extracted from within the cell. Later on, we cannot directly sequence RNA. So what will generally be done is that this RNA molecule needs to be converted into cDNA. And cDNA, which stands for complementary DNA, is the one that we can actually read based on the sequencing machine. However, the starting amount of cDNA or RNA that we extract from a cell is very little, is very limited. And most of the time it's not enough to get a very meaningful signal. So next step is to amplify. And there are two main approaches that we're going to talk about how this amplification is being done. But the idea is the same. It's just to copy these cells as much as possible until we can get a concentration that that we can get a meaningful signal from. The next step is sequencing, which, like I mentioned before, sequencing an RNA molecule means to read the RNA molecule to get some information about where the molecule comes from, what it looks like, how many copies, and so on. From this sequencing data, we obtain an expression matrix. When it comes to data analysis, especially analysis of single cell RNA sequencing data, this expression matrix would be the starting point. This expression matrix is a summary where every row represents a different gene and every column represents a different cell. And so the values of this, the value of the cells of the matrix indicate the expression of a particular gene within a particular cell. And this expression matrix is how we start is where we start the more advanced bioinformatic analysis, from which we're going to obtain our clusters, our cell lineages, and all these other signals from. Is there any question so far? Okay. Now, we're going to focus on this first part, which is the methods for cell isolation. And if you remember the previous diagram, is this part where once we have dissociated cells from the tissue bulk, we want to obtain individual cells in order to sequence them. And in this study from 2018, they highlight some different approaches to do this. So the first one, using a 96-well plate and a pipette, this is one of the pioneering methods, and I also briefly mentioned before, which is when you start with an individual well that contains your cell suspension, and you start serially diluting as you move across the plate, up to a point where the concentration of cells in the well is going to be either 1 cell or 0 cells present. So it's a little bit based on the probability, right? That for every dilution you expect a number of cells, and also, like I mentioned, it can take a lot of time. It's a lot of manual work. Second approach to do this is probably what you would imagine if you didn't have any previous background on how to isolate cells, which would be like literally picking them up by using a microscope and a capillary pipette. So with this approach, you use the capillary action of a pipette, which kind of sucks in the cells that are present in your dish. And with the help of a microscope, you can help isolate them individually. And as you can imagine, also, these first two techniques are very low throughput, and your study is most likely going to be limited to a very small amount of cells. Moving on to C, this is already a more advanced method, and one method that's still being used, that's widely used nowadays, which is based on fax sorting. The technique and the principles behind fax sorting, it can be a workshop on itself, but just to very quickly summarize it, fax sorting is the process by which you are filtering and you are dividing cells you're splitting cells based on some of their physical features, based on the type of scatter that they emit when you point a laser through them, the magnetic charge that they get on the cell based on the antibodies that bind to the surface proteins, and based on these properties, you can use other instruments in order to split them. I believe after they pass through the sorter, there will be a series of magnets that will help them split based on their charge. The fourth approach, LCM, which stands for laser capture microdissection, which is also based on a more manual approach using a microscope using a laser, where you'll be able to break apart cells from a tissue. And the principle behind this is that you have to target the laser to specific parts of the tissue where you can easily have them dissociate. This approach, though, is not widely used. At least in my experience, I have not seen studies using microdissection. So it won't be a focus on this workshop. I don't believe we're going to mention this again. Also, it's not, like I said, it's not widely used in the literature or in other studies. On the other hand, this is the most common approach used nowadays, and there are many different variations, many different approaches that use this very principle of microfluidics. The idea behind microfluidics is to using these very tiny chambers where different types of fluids are passing through in a very controlled way. So in this particular scenario, where we have this channel where a solution, a solvent carrying some microparticles is passing through and merging with another channels where your cell suspension is passing through. So the width of the channel is also designed so that you can have one cell passing at a time. And the flow of this fluid and the particles, is designed in a way that you'll have roughly one cell per particle at any given space in the fluid. And then later on, this fluid is intersected by another channel that has oil, and this oil is going to break apart this fluid into individual droplets. And these droplets, with some extent of efficiency, of course, they are going to have one microparticle and one cell within them. And that way, you can sequence and study RNA within each droplet. And that's how you get your cell-specific signal. I believe there was somebody in the bioengineering department. I don't know if that person's here today. Okay, maybe it's not present. But yeah, I just wanted to quickly mention, because I'm also a bioengineering graduate, and the design of these microfluidic chambers is a very interesting and very active area of research. And this is a very widely used application of this technology. And lastly, this other method, which the anti-EP-CAM antibody, is also a less known, less common approach to isolating single cell. And the principle behind this is also based on the binding of antibodies with magnetic particles on top of the cell surface, and then you split and you select the different cells based on the poles of magnets that they pass through. So again, at least I haven't seen studies or other technologies using this type of approach. I would like just to highlight the fact that these microfluidics and the fact sorting nowadays are the most common ways people use to isolate single cells. And we're going to expand also a little bit on microfluidics later on. Okay, I think at this point we can have a little break, a little 10 minutes break. And after that, we'll continue with the rest of the lecture.
Hi, Giovanni. Could I ask you a question?
Yes.
I'm using R from the Hoffman 2 terminal. Is that okay?
Right. Yeah, I think it should be fine. The thing is that we're going to use Markdown, and we're also going to generate some plots. So you should have some file sharing enabled so you can see the plot.
Okay.
Yeah.
Thank you.
Yep. Okay. Hopefully everybody's back. We're going to continue with the methods for cell isolation. Is there any questions so far? About anything we've covered so far? Okay. So yeah, when it comes to cell isolation, this is another figure representing also the different methods. I included it here also to get some idea of the timeline and how these methods have been evolving. So starting with 2009 with Tang et al., if you remember, this was the group that managed to sequence single cell RNA from one individual cell. And the way this was done, it was, like I mentioned, using serial dilution, which is a more manual approach to identify the cells. Later on, we also have different techniques of multiplexing, which is a similar principle, which is just based on multiplexing itself refers to the action of merging or pulling together the signal of different cells. But it was done in a way that after a series of dilutions, you'll obtain your final concentration of individual cells. And later on, we have our microfluidic approaches, which will be the following four. But with the difference that these first two technologies, integrated fluidic circuits and liquid handling robotics, these were microfluidics, but were not necessarily droplet based. This was an earlier form of microfluidics where the cells would flow around in the different chambers, and then they will try to separate the cells when some sort of detection or some sort of rate was achieved. They will be able to split them. And later on, the most advanced methods, like I mentioned in the previous slide, they will try to encapsulate the cell within individual drop. So those would be the nano droplet approaches. Or we also have pico wells, which is pretty much this setup, this little scaffold that would allow for one individual droplet to fall within each picowell. And then sequencing, RNA extraction and sequencing and so on will happen within every well. And then towards 2017, actually, we'll have the newer approaches will be in situ barcoding. We'll expand on this concept of barcoding, but it pretty much refers to labeling individual cells. And what these groups achieved over here is that they'll manage to add, through a series of permutation dilutions, they would manage to add individual labels to every cell, so that there's really not the need of physical separation anymore, because every cell will have attached to it, pretty much all the molecules within a cell will have attached to it this label, and this label indicates, hey, this RNA molecule comes from this particular cell. Yeah, and of course, and as you can imagine, this is a simpler approach and very high throughput. And the advantage is that they're able to, this way, isolate hundreds of thousands of cells very easily. At this point, I wanted to introduce another concept, or just another name, which is the doublet, which is a very important term in the droplet approach, which it simply means that, like I mentioned before, this kind of approaches, they are very efficient, but they're not 100% efficient, right? So sometimes you run into cases where you have two cells within an individual droplet, and that'll be called a doublet. So I think spelled with P, actually. Sorry about that. But yeah, it just refers to two cells being captured within the same drop. And as you can imagine, this is a problem because the whole point is to obtain the signal of an individual cell. So there are techniques. We're also going to mention some approaches of how people deal with these doublets. But it's pretty much a bad signal in your data that you have to deal with. The next step in our pipeline is the step of amplification and sequencing. And I wanted to motivate this a little bit by showing you some numbers. So for a sequencer, a sequencer requires a minimum amount of RNA, so that based on the different primers and the different reagents that they use, there has to be a minimum concentration of RNA so they can pick up the signal and start sequencing. And this minimum signal is roughly in the range of 0.1 to 1 microgram of RNA. That is required in order to run the sequencer. However, the amount of RNA that is present in an individual cell is very, very small. Typically ranges between 1 to 50 picograms. This ranges because depending on the type of cells, some cells are much bigger than others. Some cells may have a more active transcription of genes, so those factors will make the amount of RNA vary from cell to cell. And this range of 1 to 50 picograms is between 2,000 to 1 million times less the amount needed. So the problem is that we're starting with a very small amount of RNA, and we need 2,000 to 1 million times more in order to start sequencing. There is another factor that not all RNA molecules get to be captured. And this will also vary a lot depending on the type of cell isolation approach or library preparation protocol that you're using. But every one of these technologies have a certain rate associated with them. And in the case of the droplet-based technologies, they roughly manage to rescue 5 to 8% of the total RNAs that are present. Something happened here. Yeah, apologize for this. I may have deleted the rest of the sentence by accident, but this bullet point is showing is that 5% of the RNAs are mRNAs. Apologize for that, but this is an important point actually, because what we're saying is that out of the RNAs that we're capturing, not all the RNAs we're interested in. As you know, some RNAs are rRNAs, like ribosomal RNAs. Some are tRNAs. Some are non-coding, non-protein coding RNAs. So where most of the time we'll be interested only in the mRNA, which is roughly 5% of the total from all the RNAs population. So in order to deal with this problem, of course, we want to amplify the signal that we're working with. And there are two main approaches, which are, in order to amplify the signal, there are two main approaches that are the PCR and IVT. PCR, which is the polymerase chain reaction. Hopefully most of you are familiar with this. I explained briefly before, which relies on the usage of primers that will bind to your original DNA molecule, and then it will start transcribing and making copies in an exponential fashion. And then we have this other approach, which is a little older, but is still being used in the lab for different purposes, which is in vitro transcription. And what this technique relies on is on inserting your DNA of interest into a plasmid formation, into a plasmid construct, sorry. And this plasmid will have a bacterial promoter where the polymers will bind, and it will start copying your DNA of interest. And if you think about it, the PCR starts copying molecules at an exponential rate. So in a matter of 30 to 40 cycles, you're going to have millions or billions of copies of your original molecule. Whereas in vitro transcription doesn't have this exponential increase, it's more of a linear rate increase of your original molecule. So the main difference would be that the amount of copies that you can generate with each one of these methods. Another point is that generating fewer copies is not always a bad thing, because what happens when you start increasing the number of copies exponentially, this can oftentimes yield non-linearity, meaning that if originally you have two genes that you are amplifying, one gene is expressed very highly and the other one very lowly, say 10 times less than the other gene. And so if you're running an exponential amplification, of course, given the number of cycles and given the chances that you're going to pick up some of these molecules, you can end up with a different proportion, which is no longer 10 to 1. So you're losing this original ratio of the genes that you were testing. Whereas if you run an in vitro transcription, which increases the number of copies linearly, this one is more suitable if you're really interested on preserving that true proportion of the genes you're studying. I believe there is a question.
Hi, yes, I have a question. So I was wondering which isolation technology is more common, or basically most common. Currently, the droplet-based technology just capture only 5 to 8%. I was wondering whether this 5 to 8% is enough to go, or what other technology you recommend. Thank you.
Yeah, so right. So I believe the first part of your question is which technology is most common. Let me just go back a little bit. Yeah, so nowadays, given the different protocols, different technologies that people are using nowadays, is mostly based on the droplet-based technology. So this kind of isolations that use microfluidics and isolate cells on individual drops, those would be the most common approaches. And regarding the yield, the 5% to 8% of RNAs, yeah, this is relatively low, but I believe the other methods don't do much better either. I believe with the older approaches, there is like a 10% rate of, let me go back. Yeah, I believe with these older technologies, there is like a 10% rate of capture. So I can look up the exact numbers because what I'm saying now is from memory, but I believe the overall capture rate is, the difference is not that dramatic.
Yeah, okay, thank you.
Where was I? Right. Yeah, so going over the methods for amplification, we mentioned PCR and IVT. And after you have amplified your signal, the molecules are present. The next step is sequencing. And I just wanted to quickly mention that in this diagram, they used this icon for amplification, but this looks more like a sequencer machine. So I just thought it could be misleading to the reader. Anyways, sequencing. So in order to sequence the RNAs from the different cells, we're using this approach, this multiplexing approach, which simply refers to the fact of pulling together the RNAs that we're extracting from the different cells. And you may wonder, what's the point of isolating cells in the first place if eventually you're going to mix up the RNAs anyway? And the reason for that is that before doing this, we're adding a little label to all the RNAs that were captured for a particular cell. And this label that indicates the cell of origin is called a barcode. We'll go also in the demo, we'll see exactly what a barcode looks like and how it's being used. But keep in mind that what these methods are using in order to maintain the cell specificity is this little label that will be attached to every RNA molecule. And following the sequencing, now we're getting into the data analysis part. And in this part, in the diagram is showing a very simple one step from your sequencing data into your expression matrix, which is very extremely simplified, of course, because in between these two types of data, there is actually a series of steps that are taking place. So in this series of steps, what we're doing is we're converting this raw sequencing signal which is starting over here in the form of FASTQ files, we're converting these row sequencing signals into our expression matrix, which, like I mentioned before, this will be our starting point for the more advanced part of the analysis. I believe somebody put a question in the chat. I just wanted to encourage people to I prefer you raise your hand, because every time I check the chat, I'm pausing in the middle of the explanation, and a lot of times I click away and move on the slides. So yeah, it's just I prefer you raise your hand when you have a question. I'll see your hand on the screen. Yeah, so what I was mentioning here is that also the step from going from your rows sequencing reads into your expression matrix, this is a standard bioinformatics pipeline going from row sequence, from row reads to align reads, performing some quality control, and then obtaining these expression values. And these steps are the same in single cell RNA sequencing or bulk RNA sequencing. So at this point, I wanted to maybe see if we can do a show of hands. How many of you have performed any sort of RNA sequencing data analysis before? Okay. Okay, yeah, we have a few people, 6 people. And yeah, and also for those of you who want to learn about these different steps in more detail, we actually also have two workshops in the collaboratory about, I believe they are titled RNA sequencing, analysis of RNA sequencing data. And in those workshops, they will explore these steps in detail, what they mean, how to perform them, and how to obtain the signal. So at this point, I'll just very briefly explain for those of you who have no experience with this type of data, is that once you obtain your reads, these are just sequences. It would just be a sequence of letters, A, C, T, and Gs that we're generating from the sequencer. And so the first task that you have to perform is to align it to a reference genome, which will be this step over here. And in the alignment, you're trying to figure out from which gene, from which part of the genome this RNA came from. And so if you have this read that came from gene A, then you can also say gene A has one expression unit, because we have one read that mapped to it. And then the more reads you have mapping to a particular gene, then the higher your expression signal for that particular gene. Of course, it's not that simple. There are things you have to normalize. There are things you have to keep in mind. There are some technical aspects that you have to take care of. But very, very roughly, that's the idea of these steps, where you're obtaining your row sequencing data, you are assigning them to a particular gene, and then you are obtaining some expression value for that particular gene in that particular cell. And what you'll be left with is this matrix where it has dimensions n by m, where n is the number of cells that you have in the column. So cell 1, cell 2, cell 3, and your number of genes, which is your number of rows. So here you have gene 1, gene 2, gene 3, and then you have your expression values. There is a question, Ryan, or do you just not lower your hand? Okay, I think they just forgot to lower their hand. Okay, so next I wanted to talk about this feature that is incorporated into most of the single cell RNA sequencing technologies, which is called a UMI. UMI stands for unique molecular identifier. And what this means is that this is an oligo. I'm sorry. Yeah, sorry, there was a question before. For each single cell, how many reads can we get from a batch of sequencing? Oh, yeah, we'll also get to that, but that's a very good question. How many reads can we get from a single cell? And this question is actually, there is no direct answer, because the best answer is depending how much money you have, right? Because we'll talk into more detail about this, but depending on the type of sequencing and depending how many cells you want to sequence, and depending the depth, like what's your minimum requirement of sequencing depth that you have, you can get different numbers, different amounts of reads for each individual cell. The data sets I've worked with, the more shallow ones, they'll have a few thousand reads per cell. And this is very shallow signal. And it can go all the way up to a few million reads. We'll talk about smart sequencing, which this technology generates it can generate very deep sequencing libraries for individual cells. But yeah, roughly speaking, I'll give you that range from a few thousand to a few million, depending on how much money you want to invest. Okay, great question. Yeah, let's go back to the unique molecular identifier. So the UMI I was mentioning before is that these UMIs, these are little oligos, little sequences that get added to individual RNA molecules. So before we mentioned the barcodes, which is a label for every cell. And now we're mentioning UMI, which is a label for every molecule. So this implementation is actually very, I would say, revolutionary in the field, because it has allowed to obtain better quality data and to facilitate a lot of analysis. So here I highlight two uses of the unique molecular identifier, which these two aspects of the analysis are facilitated thanks to UMI. The first one is to remove PCR duplicates. So maybe here we can do a show of hands. Who has heard about PCR duplicates before? Okay, we have few people who know. So a PCR duplicate is pretty much an artifact, an error that has occurred during the amplification step. And what this error looks like is that individual molecules are overrepresented in your final library. And that is a problem, because the number of molecules is how we know the expression of a particular, the number of reads is how we know the expression of a particular gene. And if a molecule has been overrepresented in the reads, then we're artificially inflating this expression value, which is not a biological signal, it's a technical artifact. And for bulk RNA sequencing, there is an approach to remove these PCR duplicates, and the approach looks something like this, where once you obtain your reads and you align them into the genome, you are matching the reads based on their position that they align to. And so the way PCR duplicates look like is that you'll have this stack of reads, this bunch of reads that have the exact same coordinates, start and end on the genome. This is statistically is very unlikely you have this many reads stacked with the same coordinates. So most of the time, we'll just assume they are duplicates of one original molecule. So the way we'll go about this is that for this whole stack of reads, we'll just select one. And we're going to discard the rest because we just consider them artifacts that are due to some error in the PCR. However, if your data contains UMIs, actually, you don't have to do this assumption that, because they map to the same coordinates, then they must be copies. We no longer need to assume that because the molecular identifier, which is shown here in different colors, there's going to be one identifier for every original RNA molecule. And therefore, if we look at the identifier of the reads, we can see that there are, I believe, there are four different colors. There are four different identifiers, which means that these reads must have come from four different molecules. So in that sense, it makes more sense to recover 4 reads rather than just one. And as you can imagine, 4 versus 1, this is also going to affect your final gene expression values because you're getting a more accurate count based on this information. And the second approach, which is also very useful, especially when it comes to identifying variants present in the reads, is that let's look at the case of bulk RNA again, or just non-UMI technology again. As you can see, we have this bunch of reads. Let's ignore the fact that they're stacked on the same coordinates. They could or they could not be stacked like this. The point is that we have this bunch of reads, and at this point over here, we have one variant, meaning that the base at this position doesn't match the reference. And then on these other reads, we have other mismatches over here. So the way we'll go about this is that we'll identify the presence of 2 variants. We have some reads that contain variant number 1 and another read that contains variant number 2. So there must be 2 variants in this sample. However, with the use of UMIs, this can give us a more complete picture. And in order to tell which variants are true versus which ones are just artifacts. So in this case, for this variant in red, you can see that it's present in this molecule with the purple identifier. However, you see there are 4 of these molecules and the other 3 doesn't have this variant. So this variant is not consistent across the molecules. So this must be a sequencing error. On the other hand, for the second variant, you see that it's present in all 3 copies of this molecule. So it's consistent, and we are pretty sure this variant is present in this one molecule. So this one must be a true variant. And that way we can better filter false positives from our data. Is there any questions so far?
Quick question.
Uh-huh.
So how does I mean, that is very general question. This UMI works through so it reads the entire sequence of the mRNA or RNA. What if you have two genes with pretty much similar sequence, but they do have one or two single, base nucleotide that they are different? Does UMI recognize them as one, or I'm just curious to know how this UMI works. Does it read the entire sequence or just read the beginning of the mRNA?
Right. So I'm in the next few slides, in a few slides from now, I'll show a diagram where this becomes more clear. But what I can tell you for now is that these UMIs, every unique UMI will bind to a unique RNA molecule, regardless of the sequence of that RNA molecule.
I see.
Yeah, and that is done based on, it's not based on hybrid, well, there is hybridization, but it's not hybridizing to the RNA molecule itself. So it's not discriminating based on the sequence.
Mhm.
So yeah, in theory, you'll have one UMI for one unique molecule. And as long as there are two different molecules, they'll have two different UMIs, regardless of how similar they are to each other.
Interesting. I see.
Yeah.
Thank you so much.
Yeah. I believe there is one more question.
So I'm not sure if this is consistent for a UCLA sequencing core, but whenever we send cells over for single cell RNA sequencing, do they always use UMI, or do we have to do that separately?
So that depends on this is the library preparation step. So it depends how they're preparing. So how they are isolating the cells and how they're preparing the library.
Okay, thank you.
Yeah. Yeah, so these steps, just to clarify, these steps are, we are preparing the reads before reading them in the sequencer. So this is the library preparation step. And there was one more question. UMI is basically barcode, part of the barcode that is introduced during library preparation. Yeah, exactly. So during the library preparation, we'll introduce both a barcode and a UMI to your RNA molecules. Yeah, so hopefully the idea of UMI is very clear. If I were taking this workshop for credit, I would pay particular attention to this. And yeah, and moving on. Yeah, at this step, I wanted to highlight some of the different types of technologies, so the different types of library preparation and how they compare to each other and the advantages. So this is these are not all the methods, of course, there are many, many more. These are just collected in a review study. And here we have a description of how much of the transcript they identify. Now, this is something I haven't mentioned before, but what this refers to is that a lot of times what a gene will be transcribed into RNA. So that RNA is a copy of the full length of the gene. And a lot of times if we're measuring expression, for example, we don't need to know the whole sequence of the gene, right? We just need to know enough to assign the read to the gene of origin. So for that reason, a lot of technologies are not reading the full length transcript. They're only reading one end of the transcript. The RNA goes from 5' to 3'. So you see that some of these technologies, the 10x, Cell-seq2, drop-seq, they are measuring only the 3' end of the gene. And we also have this, this is an older platform that's only reading the 5'. And we have a few other technologies that they're reading the entire length, of the transcript, like SMART-seq2, for example. And here is also telling you whether these technologies are using UMI. As you can see, it's yes for most of them. The only two exceptions are SMART-seq1 and 2. So you can imagine if you don't have the UMI information, then you don't have these advantages of clearly identifying variants or clearly removing PCR duplicates. You can still get around those, but it's less efficient, and it's less accurate as if you actually had the label. And yeah, and these are the references, the studies where they publish the different protocols. At this point, maybe somebody can think of what is if we're studying single cells. So far, we only mentioned gene expression, right? And for gene expression, like I just said, you may not necessarily, you don't really need to know the full length of the transcript. You just need to know where each read came from. So you may choose to sequence one end. But can you think of what can we do if we have access to the full length of the transcript? Anybody have any idea? So having access to the full length, oh, somebody answered. Methylation. No, not necessarily, because in order to identify methylation, you need a very specialized type of sequencing. It's called bisulfite sequencing. And the principle is that there are some reagents that will identify the methylated bases specifically. So yeah, we're not dealing with any of that here. So methylation is not, it's not an option.
It can be, if you have the full length, means the mRNA is not degraded. But if you have just one of those, and the mRNA might be degraded already.
Right, yeah, that's a very good point. If we have access to the full length of the transcript, we can tell something about the RNA molecule, right? Like you said, we can tell if, for example, if it was degraded. Yeah, you can easily think of the case where you expect the gene is like very long, but you only find reads mapping to a specific part. Then, something happened to that other part. It was probably degraded. So yeah, that's a very good point. Is there any other ideas? Yeah, so other things, of course, whatever you can identify with bulk RNA sequencing, you'll also be able to identify with full length single cell RNA sequencing, which is just circles around the idea of having the full image of the transcript. Events such as if your transcript is being spliced or not, if it's using an alternative initiation or an alternative termination point, you can learn all these different features about RNA that these go beyond gene expression. These are unrelated to gene expression, but I think it's worth highlighting the fact that single cell is not limited to only gene expression. Okay. So now we're going to go into a little bit of detail into two protocols. And I wanted to start with the SMART-seq2 protocol, which, as if you remember, this is the one where you capture the full length of the transcript, but does not have UMI. So in pros, yeah, the pro is, of course, finding the full length of the transcript. And this is a outline of the library preparation. So I think it's probably, we don't need to go through all the details. So I'll just try to summarize the steps where you start with your original RNA molecule. And if it's mRNA, you know it contains a poly-A, a stretch of consecutive A's. And you make use of this poly-A in order to bind your primer that has a poly-T. So, you know, A and T base paired together. So you have your primer annealing to this part. And then once you have your primer, using a polymerase, you can copy the entire length of the RNA. This polymerase will add some non-template Cs at the end, which will allow pretty much for the synthesis of the other strand. So now you have your two-stranded cDNA copy of your poly-A, and then in the next step, this step's called tagmentation. Tagmentation pretty much just means breaking down the full transcript and adding some flanking sequence on the sides. So from your full length transcript, you're going to add these blue and these red regions, oligos. These oligos are Illumina adapters. Illumina is the sequencing, is the brand of the sequencing machine. And by adding these adapters, they're making these fragments ready to be read by the sequencing machine. And although you're breaking them apart, but you capture the full length of the transcript. It's just segmented into these different regions. And the cons of this method, of course, is that the protocol does not include a barcode. And the reason for this is because this method is not super efficient because the cells are isolated. There could be different methods for doing this. Sometimes they just do a manual isolation. Sometimes they use fax sorting. But then they're never really pulled together. They are running individual, they are using like individual experiments to sequence each cell at a time. So there is no multiplexing. And yeah, like I mentioned, there is no multiplexing. You know, we never pull together the reads from different cells. And because of that, you don't really need to label what cell they came from. And also the protocol does not, it could potentially add a UMI. And there are some technical reasons for why they don't. And of course, like we went through before, not having a UMI, it just means that finding PCR duplicates and identifying variants will be more challenging. And of course, as you can imagine, if we're preparing a library for every individual cell, then this method is pretty expensive. So this is one example where you kind of have to weigh in the pros and the cons. And depending on what you're trying to achieve in your study, you can choose to use a SMART-seq or not. So in this case, if you're only interested in gene expression, you probably don't want to use SMART-seq because it's very expensive, doesn't have these UMIs barcodes, and you don't really need the full length. So yeah, so if you're only interested in gene expression, you may want to avoid this method. And lastly, I wanted to show you this analysis that was performed where the authors evaluated the different library preparation protocols followed by sequencing. And they tried to identify pretty much which one gives a better signal. So I wanted to go a little bit over their experimental design and how they managed to evaluate these different methods. So we can just focus on this one panel where you have your cell, and within the cell, there is an endogenous RNA, meaning RNA that belongs to the cell. And you can also add some spiking mixed volume at some known concentration. So what spiking mix means is these are RNA molecules that you're adding to your experiment. And the reason for doing this is that you already know which molecules and you know how much you're adding. So you already have an expectation of how much expression you expect at the end. So this is your benchmark, and you can use spiking to test pretty much any type of RNA sequencing, library preparation, pretty much any protocol, well, not any, but a lot of protocols related to RNA. And so you have these known concentrations of RNA. So they're measuring two things, the sensitivity and the accuracy. Let me change my pointer. They're measuring two things, sensitivity and accuracy. The way they're measuring sensitivity is that given a concentration of your spiking, the ERCC molecules, you're trying to find out what is the concentration needed for you to actually identify this molecule. Eventually, you'll be able to model the data in a sigmoidal with a sigmoidal function. But I think the takeaway here is that you can see that a very sensitive method, a very sensitive protocol will be able to identify, with high probability, molecules, even if their concentration is low. So their limit of detection will be lower. Whereas if your method is not very sensitive, you'll need to input a lot of RNAs for your method to actually detect it. So your limit of detection will be high. So that's sensitivity. For accuracy, it's more straightforward. You just want to correlate the original input, how many molecules, what concentration you input in the first place, with the expression value that you calculate after the sequencing and data processing. And then you can run a correlation analysis, get an R square or an R value, the correlation coefficient, and this way measure how accurate the method is. And so starting with the accuracy, you can see that they tested quite a bunch of methods. And here they're showing this R value. So the higher the R value, the more accurate the method is. And as you can see, something you may want to remember, of course, they didn't test every method. From the methods they tested, this protocol called SUPER has the highest sensitivity. We haven't looked at this particular technology before, but we did mention other methods such as Cell RNA-seq, and this Chromium refers to the 10x protocol. So you may want to remember these three names that have the highest accuracy in detection: super, Cell-seq, and Chromium. And what this plot is showing is pretty much this accuracy, the R value, how it changes for different number of reads. So we don't need to dig too much into the details. I think you just need to know that with more reads, of course, every method is going to improve accuracy. And you can see that around these lines, I believe is the first line where these curves start to plateau, meaning that no matter how much more you increase the number of reads, it won't cause a big change in the accuracy. And among the least accurate technologies, we have MARS-seq, GNT. We haven't mentioned this one also. We have the method developed by Tang et al. I believe this one refers to their 2009 paper. And we have SMART-seq 2. Now, in terms of sensitivity, if you remember, we're measuring it based on the detection limit. So in this case, the smaller this M value, the more sensitive your method is going to be. And just to reiterate, because that means you need less concentration in order to identify a given molecule. So based on this analysis, we see that this technology, Cell-seq 2, STRT-seq, SMART-TER, they have the best detection values. Cell-seq 2, STRT-seq, SMART-TER. And up here we have other technologies that are less sensitive. We see super here, which, if you remember, is the most accurate, but as you can see, it sacrifices some sensitivity in order to get high accuracy. We have MARS-seq, GNT, and GEMCode. And SMART-seq 2 is also up here. Yeah, so I think at this point, it would be good to take a 10-minute break. And I'll leave this slide on. I think you should, I think it will be good you take some notes of the best performing methods. And yeah, after 10 minutes, we're going to have a little quiz. Don't worry, it's not for your grades, just an activity. And yeah, and in the meantime, if you have, feel free to write in the chat. Now I can look at your questions with more time. And we'll see each other in 10 minutes. Okay, at this point, oh, sorry, before that, I wanted to, somebody asked a question during the 10-minute break. The question was whether if, let me go back to the previous slide. Yeah, regarding this table, if you only sequence either the 3' or 5' end of a transcript, how can you tell which gene is coming from? Like, don't you need the full length transcript in order to identify the gene? And so the short answer is that no, because when you sequence one end, you're sequencing sometimes 75, sometimes 100, sometimes even more 150 bases of the end of the transcript. And those 75 to 150 bases, those are long enough in order to identify the gene they came from. And also because of the current limitations for Illumina sequencing, or for the sequencing given this type of library preparation, we're limited to 100. I think the maximum is like close to 200 bases. That's why, you know, it needs to be, even when you sequence the full transcript, you still need to break it down into smaller pieces. Yeah, so final answer is that as long as your read is long enough, no matter if it came from the 5', the middle, or the end of the gene, if it's long enough, then we should be able to find what gene it came from. Okay. Thanks for that question. So now I wanted to try to have, oh, sorry. Okay, I wanted to do this online quiz, Kahoot. So for this, I'm gonna ask you to please go to this website, and it will be better if you can do it from another device, not the computer you're using. Just so, because you're also going to be looking at this screen. So if you have a cell phone, please go to Kahoot. And let's see. And now I'm going to generate a PIN number, which you can see over here. You'll need to enter this number into your into the website. Cool, we have our first players. Yeah, you can put any username you want. Let's see, I guess I should be expecting around 20 players. Is anybody still struggling to get into the game? Yeah, you have questions, or how can we help you? I think there are some people raise their hand. YJ? Okay. Yeah, let's move on. So I'm going to show the first question, and then it will be multiple selection. Oh, sorry, there are messages in the chat. Okay. Yeah, so it will show you questions. It will be multiple choice and you select the best answer. And let's start. So first question, what is the normal amount of RNA in a cell? And we mentioned this in the slides about amplification, right? And how little RNA was present in a typical cell. And yeah, just to clarify, this refers to total RNA, so like the amount. Okay, yeah, most people got it right. Yes, the amount is 1 picogram. Well, like I said, it ranges from 1 to 50 picograms, which is 2,000 to 1,000,000 times what we actually need. Great. Let's go to, these are the rankings. Congratulations. And we'll go to the next question. What is multiplexing in single cell RNA-seq? Yes, again, choose the best answer, just to keep them short. They are not very complete, but select the ones you consider correct. See. Yeah, so I actually put 2 right answers. The idea refers to the process of pulling together the signal, right? So it starts off by pulling together different cells, and what we end up with is a pool of all the reads coming together from these different cells. The multiplexing is just this idea of merging them together. Great, 4 people got it right. Are people running out of time, or it feels like a little quick? Can anybody raise their hand if they feel like they didn't have enough time? YJ? Oh, yeah, there is a time limit of 20 seconds. I'm just saying that it feels shorter than that. Okay, great. Let's move on. Okay, great. We still have Anne, first place, followed by Anna. It moves to correct answer as soon as everyone answers. Oh, really? Right. Oh, yeah, of course. If everybody answered, then yeah, the time completes. Okay, let's go to our next question. Which one is the most recent technology? So I have 4 options. Which one is the most modern? Great, yeah, most people got it right. 10x, yeah, 10x developed close to 2017. And like I mentioned, it's probably one of the most commonly used technologies in single cell nowadays. Yeah, SmartSeq, on the other hand, not so recent, 2014. Yeah, 2014. And CellSeq and StarSeq is the oldest, so I'm glad nobody selected that one. Great. Great. Let's go to the next question. What is a doublet? Yeah, this was mentioned during the cell isolation protocol. The cell isolation technology, sorry. Great, yeah. Vast majority got it right. Yeah, it's 2 cells captured in the same droplet. Of course, that's an artifact. That's something we don't want, and that's something we want to get rid of during the data analysis part. Yes. Great, the top 2 participants still there. Let's go to our next question. Which one is not an application of single-cell RNA sequencing? Great. Yeah, once again, most people got it right. And to increase gene expression signal is not an application, quite the opposite. Generally, you end up with a much diminished signal with single cell technology. One of the main limitations of this type of analysis, this type of data. Great. Cool, the podium remains unchanged. Let's move on. What is the purpose of UMIs? So a very important feature in single cell RNA sequencing technology. Yeah, once again, most people got it correct. Not to be confused with barcodes. Actually, I shouldn't explain too much. I believe that's the next question. But yeah, great to the people that, congrats to the people that got it correct. Let's move on. Yeah, next question. What is a barcode? How is it different from a UMI? Great, yeah. A label to identify an individual cell. So this is what I was about to explain in the previous question. I didn't want to give too much away too soon. Barcode to identify individual cells, UMI to identify individual molecules. It's important you keep, you understand those two different concepts. Cool. Let's move on to the next question. What percentage of RNA is captured inside a droplet? So once again, going to the slides, I was showing some numbers. I believe when talking about amplification. And yeah, most people got it right, 5 to 8%. Cool, still remain the same podium. Let's go to question number 9. Which of these technologies sequence the entire length of the transcript? Yeah, also it was mentioned just before this quiz. Hopefully it's still fresh in everybody's memory. Yeah, SMART-seq2, probably the biggest advantage of this technology. Let's go to the next question. And the last one, out of the methods tested by Svensson, that's the last slide I show. Which one had the highest sensitivity and specificity? So first method for highest sensitivity and second for highest specificity. Yeah, this question was a little harder. It requires some memorization from the older slide. But most people still got, oh, sorry, no, not most people, 4 people got the right answer. Cell-seq 2 had the highest sensitivity, while super-seq had the highest specificity or accuracy. Yeah. And our podium, we have Chris H, Anna, and the winner is Anne. Congrats, you just won nothing really. Everybody can clap for you guys. But yeah, I'll make sure to include this kind of activities for all 3 sessions in order to just review on the material and also make it a little less monotone. Anybody had a question? YJ? Okay. Great. Great. Now going back to the slides. Yeah, so after the little review of the material, I thought I wanted to, it would be good to actually look at the data, actually look at the output you get from the sequencer. And so you get an idea of what we're working with. So in order to do this, we're going to focus on yet another technology. We already talked about Smart-seq2, and now I wanted to talk about 10x. So 10x Genomics, in reality, is the name of the company. The protocol is, I believe it's called, the full name is 10x Genomics Chromium for single cell RNA sequencing analysis. And the type of technology that is being used here is based on barcoded gel beads. We mentioned this, a similar example actually, when we were talking about the microfluidic technology. And I just wanted to, well, I guess at this point is to repeat once again the mechanism that is being used with microfluidics, where these beads are already prepared, and these beads are meant to be captured inside a drop with one cell. So the way that it works is that the beads will pass through this through this channel, and they will bind. They will couple together. They will get together with a cell, and then by adding oil into this fluid, we'll be able to split them into individual droplets, where ideally one droplet will contain the bead and the cell and one cell. And at this point, I just wanted to show why do we need a bead in the first place. The reason for that is that these beads, they have these oligo sequences attached to them. So in this particular bead, you can see there is a sequence, there is a DNA oligo that is bound to the bead. And this oligo contains different features. The blue part over here is a poly-DT, it's a poly-T sequence, followed by a UMI sequence, and then followed by the 10x barcode. And lastly, this R1 sequence I'll show what it's used for. It's used for the amplification step. But I wanted to highlight is that you see that at this step, you have individual bits captured with individual cells. So you can see that every bit will have thousands. Actually, I'm not sure of the exact number. I believe it should be several thousands of these oligos. But all the oligos will have the same barcode. But different oligos will have different UMIs. So, and this is because every one of these oligos is meant to capture an individual RNA molecule that came from that particular cell. So, and we're assigning the cell's label here in the barcode. So this is the mechanism by which we're able to implement a UMI, this identity to every molecule and for every cell that we're sequencing. Later, you collect these droplets that contain the single cell gems. The gem just means the droplet with the cell and the bead. And later you perform the reverse transcription step, which will take place still inside every gem. And this reverse transcription, of course, is the part where you are converting your RNA into cDNA. And then later you'll remove the oil so that by removing the oil, you're pretty much just merging together all these droplets because they're all solvents. So they will just dilute into one solution that contains the sequence from all the individual gems. This will be the multiplexing part when we're pulling them together again after already labeling, thanks to the beads. And from this step forward, we'll go ahead and amplify the signal, make copies, and then sequence, like reading the actual cDNA molecule. Is there any question about this? Okay. The next slide. The next slide goes, this is a little bit of detail about what I just mentioned. We saw the library preparation protocol for SMART-seq2. Here we have the protocol. This is just a more detailed description of how we go from capturing the RNA all the way into generating a library that's ready to be sequenced. So same way as SMART-seq2, see, we rely on a poly-T sequence that this will capture the poly-A sequence that the messenger RNA should contain at the end. And you can think of this step as the fishing step, like the capturing step. And once you have captured the molecule, it's just a matter of copying this strand, transcribing this strand, adding some primer on the other end. So you can, this part is called template switching. So you'll sequence the other strand as well. And in the process of doing this, you are incorporating this sequence that was present in the bead oligo. You're annealing it, you're joining it together with the sequence of the RNA. as you can see, represented by this thin line. And then altogether, this very long sequence will contain your barcode, your UMI, it will contain the poly T that was used to capture the RNA. It will contain the RNA sequence itself. And these read1 and read2 are regions that are used for amplification. So your PCR primers will bind to these read1 and read2 parts. And the P5 and P7 sequences are sequences that are used for the sequencer. Actually, I have the full explanation here. Yeah, so here we have our barcode, our UMI, and P5, P7, and also this very short sequence called the sample index, which is just, you can think of it as an extra label to identify the sample that you are analyzing. Sample in this case, of course, would be defined based on your experimental design. Maybe you have single cells from person 1, so that will be one sample versus single cells of person number 2. Yeah, so it's just an extra sample that you are incorporating into your long construct. And just as a little exercise, here I included the length of each one of these sequences. So your barcode, in the traditional protocol, of course, there are special protocols where they would change these parameters, but the most commonly used protocol, your barcode will be 16 nucleotides long. Your UMI will be 10 nucleotides long. The sample index will be 8. And these P5, P7 sequences, we're not going to use, they're only used for sequencing, but we don't use them downstream. They're around 20 something bases, but it won't matter to our feature analysis. And the reason why I'm including their length is that you can also calculate the number of unique barcodes or unique UMIs that you can synthesize. And the way you calculate that number would be with 4 to the power of n, n being the length. So for example, how many unique barcodes can we generate? That would be 4 to the power of 16, which will be, of course, a very large number, I think in the billions. And UMI, how many UMIs can we generate? That will be 4 to the power of 10. That number is a little over 1 million. So yeah, so this length will give you an idea of how many unique sequences you can generate. Are there any questions? Okay, and yeah, and in the final step, after you've prepared this construct and you sequence them, the sequencer will spit out 3 reads, not to be confused with the PCR read, but we'll just call them R1, R2, and I1. These are the pieces of information that we're getting from the sequencer. And then our R1 would only sequence these 2 fragments, which is the barcode and the UMI. Our R2 will sequence the actual sequence of interest, the sequence of the RNA we just captured. And then our I1 sequence would be the sample index, which is only 8 bases, and will only give us information about what sample this is coming from. So if you see the logic here, what we're doing, what this technology is doing is reading what we wanted to read in the first place, which is the RNA molecule. But then it's also adding this information, the barcode and the sample index. these reads are linked together. So for every RNA molecule, we're also going to know which cell it came from and whether it's a unique molecule or if there are duplicates or something else. And here, I wanted to just share, my terminal. Are you guys familiar with Huffman? Have a show of hands. Okay, we have some people. Okay, 5 people. So Huffman is this computer cluster in here at UCLA. It's pretty much this, you can think of it as a collection of computing resources that you can log into, and then you can run a program or a script that requires, a lot of resources, high memory, or needs to be running for a long time. And here on my Huffman account, I have downloaded some 10x data. So here I'm just going to show, let me actually share my whole desktop. No, so yeah, somebody asked if they need to run Huffman. No, don't worry, you don't need to run Huffman. You don't need to do anything at this point. I'm just going to show you the data. So you don't need to follow these steps on your computer. So I just wanted to remind you what we're looking at. We're going to look what these 3 reads that we obtained from 10x look like. Like I mentioned, read 1 with the barcode UMI, read 2 with the actual insert, the actual RNA, and read 3 or what I1 with the sample index. So here I downloaded a publicly available data set. And here we have our, we have 6 files because it's 2 technical replicates. Each replicate will have its own R1, I1, and R2. And you can tell the replicate label here on the L001 and L002. So let's just take a quick look at the read number 1. For replicate number 1, let's take a look at R1. And this is a FASTQ file format. You don't need to know what every line or everything means. I'm just going to highlight this every 4 rows. This is going to be a different read that you obtain. And these reads are 26 bases long. 26 because the first 16 contain your cell barcode and the next 10 contain your UMI. And every read has its own name, which is shown here. And yeah, so that's the information that we're capturing with our read number 1. If we look at our read number 2, you can see these reads are longer. So these reads are 98, I believe 98 bases long. And this sequence is actually the sequence of the mRNA that we're trying to capture, that we're trying to analyze. So this sequence will actually match one gene in our reference genome. And lastly, we have our I1, which, like I mentioned, contains only the index sample. So it's very short, it's only 8 bases long. And this will give us information about which sample it came from. For this particular data set, I believe they're all the same. There might be some mismatches present, but they're all coming from the same sample. And yeah, and the way they are linked together is just that they have the same name. A particular read will have the same name on all 3 files. So hopefully this won't take very long because these are very big files. Yeah, people have questions. Jenny? Or
Sorry, I just had my hand raised that I was familiar with Hoffman too still. Sorry about that.
oh, okay. Yeah. And Shabnil? You have a question? Oh, somebody's asking if R1 means reverse and I1 means index. I1 means index, yeah, the sample index. R1 doesn't mean, that's a good question because it doesn't mean reverse, it means read, but it is, it could be reverse. And this is probably a little confusing right now. If you have experience with RNA sequencing data, you know that your reads could even match, the sense of the RNA that you're trying to sequence, or it could align to the reverse sequence, the reverse complement. So that's why it could be confusing, but in this case, R just means read. And I think it should be done running. So what I did here is that for this particular replicate, I searched this read name, and then I printed the following 3 lines. So this first, this read name is present. This was found in the I1 file, and we can see it has the 8 bases sample index sequence. This was found in the R1 file, so it contains our 26 bases with the barcode and UMI. And this was found in the R2 file and contains our 98 bases of the target RNA. And yeah, and this, because they have the same read name, they are referring to the same construct, which is this construct over here. And that's how we can link together and say, this RNA molecule belongs to this particular cell. The name of the cell will just be the barcode. And this is its own identifier. So somebody replied that the R1 and R2, R1 is forward and R2 is reverse. Yes, that's true. Because if you look at the diagram, you have a 2-stranded construct, right? So in one of them, you're going to be reading in the right direction, and in the other one, you're going to be reading in the opposite direction. So yeah, they summarize it really well in the answer. So thanks for that, Qingwen. So yeah, when you have per n reads, usually one read will be forward in the same direction as the RNA molecule, and the other one will be reverse. Shabnil? Okay. And I still had a couple more slides to go through, but I think, yeah, I think it will be good to stop here because they will require a bit of explanation. And yeah, and tomorrow we're gonna pick up at this part, and then we're going to continue with the proposed program. Yeah, thanks everybody for attending, and I'll see you tomorrow. You can stay if you have any questions, and we're going to end the recording here.